Good morning, students. So today in this video, we will be discussing the very introduction of your cancer genetics. So, what is the genetic nature of cancer? Now, cancer is a complex disease, right? And you know that cancer basically results from the process of uncontrolled growth and division. Now the cell, the total number of cells in our body needs to be constant, right? That means the total number of dying cells will be equal to the total number of growing cells. But we have to focus that cancer is a cell proliferation, which results in a mass that invades your neighboring tissue, and ultimately it moves to different parts of your body, resulting in metastasis. That means the cell, due to its uncontrolled growth and division, loses its confinement and moves to different distant parts of the body. Now, in order for a normal cell to transfer into a cancer cell, there are certain genes which regulate it. So genes which regulate the growth and differentiation of the cell, the genes which regulate the total number of cells in our body. So these genes basically malfunctions. These genes basically undergoes mutation. Now here I want to just focus on one point, that you know that we all know what is mutation, right? And this mutation is change in the genetic sequences, which results in severe consequences sometimes. So when this particular genetic mutation occurs, usually mutation can occur in one particular gene, right? That is monogenetic, right? Monogenetic mutation. But when there are polygenetic mutation, multiple genetic lesions at a time, it becomes very difficult for the body to cope with this situation, and that basically leads to what we call cancer. So when normal regulation is altered, uncontrolled growth is initiated, and a malignant tumor develops. So as I will move on to the videos, you will be understanding that what is your benign tumor and what is your malignant tumor. So the genetic changes can occur at many levels and may lead to gain and loss of entire function of the chromosome, right? So we will also be discussing that what is this entire chromosomal mutation which leads to gain in function and which leads to loss in function, and ultimately both basically results in cancer. Now if we move on to carcinogenesis, carcinogenesis is a multi-step process. It is not a single step process, okay? And at both the phenotype and genetic level, they basically are produced by accumulation of multiple mutation, which we have talked before, that it is not due to mutation of one gene, rather it is mutation of multiple different kinds of genes at a time, which results in this type of disease. So it is initiated first. So it is initiated by a chemical molecule, and that is your carcinogen, the carcinogen which basically causes your cancer. Now this carcinogen results in your genetic change. So the mutation which I am talking about is related to your carcinogen. So this carcinogen basically brings about your genetic change, and that will result in the promotion, the altered cell, right? This altered cell, this promotion is converted to, is then moved to transformation. Now what is this transformation? First, initially there is a cell multiplication. And as you know, when the cell starts to multiply, there is more and more number of mutation. That is why I am saying that it is multiple genetic lesion at a time. So they are saying genetic change. And that leads to malignant cells. So slowly the cell becomes malignant. Slowly the cell starts to lose its confinement. And ultimately when the genetic changes further occurs, it results in a formation of your malignant tumor. So basically the carcinogenesis comprising of initiation, promotion, transformation and progression. So initiation is from your carcinogen to promotion, that is the first genetic change. Then there are multiple genetic change which will lead to transformation from cell, number of cells to malignant cell, and then to malignant tumor. So next is the tumors are clonal. This is very important. Clone is the similar kind of cells. You have learned in your immunology that what is clonal selection. If one particular antibody, a cell containing antibody is selected, and that basically clones to recognize the antigen. So that is clonal. So a tumor is formed by the clonal expansion of a precursor cell that incurred genetic damage. So this particular line is very important. This particular sentence is very important. Genetic damage makes one cell, and that particular cell is a precursor cell. So this precursor cell undergoes clonal expansion. So it will undergo expansion. Even though most malignant tumors are monoclonal in origin, so they are single type of clones. By the time they become clinically evident, their constituent cells are heterogeneous. So their constituent cells are not normal. So you know that if this is a normal cell, so if I say this is a normal cell, so the first mutation has undergone a change in this cell, right? Then as the cell multiplies, there is a second mutation. Then there is a third mutation. Then there is a fourth mutation and later mutation. So in every step, you get to see mutation. And all this mutation leads to the formation of this malignant cell. So, you know, so from this particular slide or this particular part, we understand that a cancer is basically the consequence of multiple mutation at a time, multiple genetic lesion at a time. So normal gene therapy and all those therapies can treat a monogenetic disorder, right? But a lesion, multiple lesion like cancer is very difficult to be treated, and more advanced technique is required. So cancer arises from the accumulation of genetic aberration in somatic cell. So again, I will come to tell you that somatic cell and germline cell. So the normal somatic cell undergoes genetic aberration, and this aberration consists of mutation and multiple chromosomal defects, right? Together, they will lead to, and this is very important, altered gene expression. So those genes which was expressing a normal set of protein, having a normal phenotype, they will start to express something different. And so that different phenotype is basically lethal to the human. So over 500 genes are now known to be involved in cancer development. So first you have to chalk out those 500 genes, right? And then to think about the treatment. So this 500 genes and many other genes are involved in the cancer, so that needs to be chalked out first. So the basic mechanism of all cancer is your mutation. Mutation is a change in DNA. So it says that germline mutation are responsible for 5% to 10% of cancer cases. And it is also called familial cancer. So the one which is inherited in family. And this mutation are present in every cell of the body and are passed from parent to child. Those are familial mutation, and genetic counseling is very, very important to prevent those transmission of this disease. But sporadic cancer or somatic mutation are caused all of a sudden. It is acquired, and it is being caused by not only tobacco but also different kind of carcinogens, which may be physical, which may be viral, right? UV radiation is one of the carcinogen. Various toxic chemicals can be carcinogen. Even virus, oncogenic viruses can also cause cancer. So cancer tends to involve multiple mutation. This is what I am trying to say. Mutation inactivates suppressor gene. So this is one question. What is a suppressor gene? Suppressor gene means that suppress the cancer. You know, tumor suppressor gene that suppresses the tumor, right? Cell proliferates. Mutation inactivates even the DNA repair gene. So those which can repair the DNA, they can also not function. Proto-oncogenes then mutate to oncogenes. So we'll come to know in our later slide that what is proto-oncogenes and how it is being activated to oncogenes. And more mutation, more genetic instability, and leads to metastasis. So you get to see in this diagram that benign tumor, right? They are actually confined. And then slowly they increase in number and they undergo malignancy and they start to spread from the confinement. Then through the systemic pathway, they move to distant part of the body, causing multiple tumors at multiple sites, and that is your metastasis. So chromosomal defect, you know that we will be discussing later, this translocation, inversion, insertion. These are all your different kinds of mutation. But before that, I will focus on loss or gain of whole chromosome, loss or gain of whole chromosome set. So, this is your loss of functional mutation. In the very beginning of the slide, I have talked about loss of functional mutation. So, this is a tumor suppressor gene, right? So, this tumor suppressor gene basically stops the cell cycle at different checkpoints. We have already discussed, right? So, it is having checkpoints where the DNA is being rectified and then the DNA moves. But if you have mutation, it loses its function. So, what it does, what function it is having, that is the tumor suppressor, that particular function is lost. So it can't prevent cancer. So it will produce cancer. So that is your loss of functional mutation. The function of tumor suppressor gene is basically lost, which was used to stop the cell cycle. So the cell cycle will not stop even if the DNA is being damaged, and that will basically lead to cancer. Again, proto-oncogene. This is being used for normal regulated cell growth. But if due to mutation it gains its function, what will happen? It will lead to the production of oncogene, and this oncogene will lead to cancer. Oncogenic genes are responsible for cancer, but that can only be activated, gain its function by mutation of proto-oncogene. So this is the loss of function and gain of functional mutation. Apart from it, types of proto-oncogenes are cellular oncogenes, proto-oncogenes which have been to mutate in any individual, that is your cellular oncogenes, and normal oncogene, N-oncogene, proto-oncogenes that have not been found to mutate. So if you move on to the mutation status of your oncogene, C-oncogene and N-oncogene, C-oncogene is basically found to mutate in every individual, but N-oncogene is not being able to mutate. So this is basically the overall slides. So this is basically the overall process of your cancer. So if you have any questions, so if you go to all the slides, I will just give a brief recapitulation of the whole. That from the first slide, we know that cancer is a complex disorder and it leads to cell proliferation, right? Next is, carcinogenesis. So what is carcinogenesis? How it is being initiated by your carcinogen, and how promotion, transformation, progression leads to the proper establishment of your cancer. Then tumors are clonal. So what do you mean by clonal? That means clonability of your tumors, how they clone and they form one type of cell which have lost the whole proliferation process. Next is the mutation. So the mutation, the single mutation and multiple mutation, which basically brings about your cancer. And then these are basically the different mechanism. You get to see in this mechanism how this benign tumor has basically been converted to metastasized tumor. It has invaded the cell and the tissue to different parts, bringing the metastasis. And this has happened when there is more genetic mutation, more genetic instability, and metastasis disease. And this is basically the chromosome defect. The chromosome defect can be structural and numerical. So in structural, when the structure is manipulated, right, the nucleotides are manipulated. Suppose a nucleotide has entered, has been added, insertion. If it is being duplicated, duplication. If it has been deleted, then deletion. So all will lead to change in the amino acid sequence and may have lethal effect. So this is your loss of function and gain of function mutation and how they bring about cancer, right? Then the proto-oncogenes and its two types, that is C-oncogenes and your N-oncogenes. And this will end the class today. So in the next class, we will be discussing the other different genetics of cancer and how the gene regulation brings about the regulation of the cancer, expression of the cancer, and how it can be prevented. Thank you. If you have any queries, please comment on Google Classroom. We will discuss.